Hi everyone, I hope everybody's doing fine and looking forward to getting back into things, even if it's in a different format than what we're normally used to. This is different for me as well, but we'll survive and make it to the end of this semester, I'm sure. This first week, what we're going to do is going to be looking at something that hopefully is familiar to most of you all. We're going to be talking about mitosis, which is something that I bet you guys did in high school or sometime in the past. Mitosis is cell division. Cell division, which is responsible for three basic processes: for cell growth, for repair, and replacement of worn out cell parts. The important thing to remember in mitosis is one cell undergoes the process of mitosis and produces two cells that are identical to the parent, identical in all of its genetic makeup and basically its function. Thing to remember also is that everyone basically began life as a single cell zygote. You remember that from before. And that single cell zygote has undergone billions of mitoses to produce the organisms that we are today. However, one of the things that's interesting is not all cells undergo cell division in the same process. You remember that we've got two big different categories of cells. We've got prokaryotic cells in organisms that are in the domain archaea and bacteria, and eukaryotic cells, those cells which have a nucleus, which is in the domain eukarya. Prokaryotic cells do cell division in a completely different way. They are much simpler than eukaryotic cells and generally have only one chromosome. And what happens in prokaryotic cells is prokaryotic cells undergo division by duplicating that chromosome. It makes two copies of that chromosome. The chromosome then gets attached to the membrane. And after it's attached to the membrane, the cell simply divides in half. And where formerly was one cell, now we have two. The process of prokaryotic cells undergoing cell division is called binary fission. It is not mitosis. Only eukaryotic cells are the cells that undergo mitosis. Eukaryotic cells are much more complex. They usually have many chromosomes that are present within their nuclei, and they go through many different steps of the process of mitosis, which is what we're going to talk about. Before we do that, what we're going to do is we're going to look more at the nature of the chromosomes and see what happens when the cell is getting ready to undergo division. Okay, looking at the nature of chromosomes, chromosomes contain your genetic material, which is found in a DNA molecule, but chromosomes aren't just made up of DNA. Chromosomes basically are built of a material that's called chromatin. Chromatin is a combination of DNA, but also protein. So chromatin is a combination of DNA and protein. It works like this. The little protein that makes up chromatin is in small little spheres, and those small little spheres are called histone proteins. So histone proteins, and there's many of them. Surrounding those histone proteins is a coil of DNA. So here is the DNA that coils around those histone proteins, and there's many of those along the strand of the chromosome. Surprisingly enough, there's more protein in a chromosome than there is DNA. About 60% of a chromosome is built out of those histone proteins, and only 40% is built out of the DNA molecule. Another thing is that the chromatin can exist really in two different forms. Chromatin can be in a form that's actually visible as a chromosome, or it can be in a form that's really difficult to see because it's not condensed. So chromatin can be in a form that's called uncondensed, and if it's uncondensed, it's not visible. Most of the time when you look at a cell and when you look at a nucleus, the chromosomes or the chromatin is uncondensed. Only when the cell gets ready to do mitosis, the chromatin is able to condense, and it condenses into a visible form that we call a chromosome. It's still chromatin. It can be condensed, it can be uncondensed. When it's condensed, we call it a chromosome because it's visible. When it's uncondensed, it's just chromatin because we are not able to see the actual structure of the chromosome. Here, this diagram shows it a little bit better. Here is an illustration of a DNA molecule. You can kind of see the double helix there. And the DNA molecule is now wrapped around those histone proteins like we saw in that other diagram that I drew. And then that whole business can coil up even more and ultimately form the visible condensed chromosome that you see here on the right-hand side of the drawing. One of the things that I didn't mention before was that there's a specific term for the grouping of histone proteins and a wrapping of DNA. So here is one of those structures where we have some histone proteins and a strand of DNA wrapped around it. That specific structure is known as a nucleosome. So a nucleosome is a grouping of histone proteins with a wrapping of DNA around it. So we've talked about the nature of chromosomes. We've seen how they're built, what they're made up of, and we've talked about how chromatin can be either condensed or uncondensed. There's another thing that we need to spend some time on since we're dealing with cell division. We need to see that chromosomes themselves can exist in two different forms. Chromosomes can either be unduplicated or they can be duplicated chromosomes. Most of the time when the cell is just in its doing its job and it's not undergoing cell division, the chromosomes are unduplicated. However, when the cell gets ready to do mitosis, we've got to make copies of those chromosomes so that we can divide those copies into the new daughter cells. At that point, those chromosomes become duplicated. So here we have one unduplicated chromosome. This is an unduplicated chromosome. It's in a cell that's doing its metabolism. It's just doing all of its work that it's designed to do. It's not undergoing division. But if that cell gets ready to do cell division, it has got to make a copy of that chromosome so that it can send those copies to the daughter cells that it's getting ready to produce. When the cell does that, it goes from a single unduplicated chromosome to a duplicated chromosome. So this is one unduplicated chromosome. This is one duplicated chromosome. So this is one duplicated chromosome. And when the duplicated chromosome occurs, the copies happen to stay together for a while. So these copies and the duplicated chromosome are called chromatids. So here are two chromatids. So we have two chromatids. The chromatids get connected, and the connection where the chromatids are held together, this is called the centromere. So a duplicated chromosome is built out of two chromatids connected by a centromere. I say this over and over, two chromatids connected by a centromere. A couple of important things to remember. Chromatids are only called chromatids when they're with their partner. There is no such thing as one chromatid. Okay? Chromatids only exist as pairs. So you can only talk about chromatids when they are together connected by a centromere. The other thing that's interesting is this. This is considered only one chromosome. That's one chromosome that's unduplicated. This over here is considered one chromosome, but it's one chromosome that is duplicated. Okay? So, always remember, chromatids are only chromatids when they are connected with their partner. And chromosomes can either be duplicated or unduplicated. So here in this diagram, you see that here is one unduplicated chromosome here on the left. And the cell has gotten ready to undergo mitosis. It has copied that chromosome. Now we have one duplicated chromosome here on the right. We've got the chromatids, which often are called sister chromatids, that are connected by a centromere. Putting all that information together, you can see here in this diagram there is a duplicated chromosome on the left, built out of 2 chromatids. And if we zoom in closer, we can see the histone proteins with the wrapping of DNA forming a nucleosome. And the whole thing is built out of that material which is known as chromatin. This diagram here is known as a karyotype, and looking at this is a karyotype of a human chromosomes, and these cells or these chromosomes are from cells that are getting ready to divide, so the chromosomes are duplicated. You can see the chromatids and the centromeres and all of these. If you kind of look through those, there's 46 chromosomes in there. There's 23 pairs, and what normally happens is when this is done, the pairs are paired up with one another because they look alike, and we'll see what that looks like in the next picture. So this is a human karyotype where the chromosomes have all been paired with their partner. And you can see that there are 23 pairs plus the sex chromosomes down here. This is obviously a male with an X and a Y chromosome. That concludes what we're going to be talking about as far as the structure of chromosomes. For the next PowerPoint, we're going to be looking at the cell cycle and the phases of mitosis.